Imagine trying to reach someone in a locked room, but you can't because every door is locked shut. That's essentially what happens when we try to treat multiple sclerosis, otherwise known as MS, inside the brain. In MS, the body's immune system will accidentally attack protective coating around nerve cells, treating it as a threat. Once that coating, known as myelin, is damaged, communication between the brain and the body will become weaker and slower. And although we do currently have MS treatments that try to prevent the progression of that damage, they all face one major obstacle, which is crossing the blood-brain barrier. This barrier acts like a security gate that protects the brain. It's good at keeping dangerous things out, but it also blocks most medicines from getting in. So even when we have drugs that could help, they oftentimes can't reach the parts of the brain where damage is actually directly happening. Our project proposes a tiny nanoparticle that can carry treatment directly into the brain and release it only where multiple sclerosis damage is occurring. This nanoparticle has 3 key features, which you can see on the slide. First, we have a passport to cross the blood-brain barrier, which you'll see as these blue dots on the surface. They help the nanoparticle pass through the barrier safely, the same way that many nutrients already approved to pass through would actually enter the brain. This would allow the medicine to no longer be stuck outside the brain and actually reach the tissue that needs it. We also have medicine inside the particle that stays locked until it finds damage. You can see these as the yellow dots. This medicine will stay locked during circulation and will only release where there is this damage occurring as well. Then we have an unlocking system that works with the medicine, which is triggered by inflammation at sites of multiple sclerosis. So once a nanoparticle enters an area affected by MS, it'll encounter something called ROS, reactive oxygen species, which is something that is released during inflammation. These high levels of ROS will act like a key that unlocks a treatment, and the particle will open only if these inflamed regions have these elevated levels. To test whether the strategy works, we ask 2 important questions. Does it get into the brain, and does the medicine release only at the damaged site? To test this, we propose a standard mouse model, and simply put, we will track whether the nanoparticle actually crosses the blood-brain barrier using fluorescent labeling and brain imaging, and then we will test whether inflammation truly triggers the particle to unlock by measuring how much medicine is released when we have these ROS levels and when we don't. If this approach works, it could solve one of the biggest challenges in MS treatment, getting therapy into the brain in a safe and targeted way. So in summary, our project delivers medicine to the brain using a simple 3-step idea. First, it crosses the barrier, then it detects damage, and finally, it releases a treatment that prevents the progression of multiple sclerosis. Thank you.